And can you see my slides?
I see it.
Perfect. Okay, great. So today I'll be sharing some work that we've done regarding the validation of a new assay for rapid detection of viable salmonella in meat and fresh produce. So just to first off, start up a little bit about the Canadian Research Institute for Food Safety, where I am and direct here at Guelph. So this is a dedicated building for the study of foodborne pathogens. It includes specialized laboratories such as the CL2+ laboratory that we're in the process of upgrading to a CL3, a meat processing or BSL3 for those elsewhere, a meat processing pilot plant. We have gastrointestinal models, human gastrointestinal models that we use to study microbiome, and various other facilities, including dedicated genomics analysis and bioinformatics laboratories. In my lab, I conduct studies based on phenogenomics, which is the combination of phenotype and genotype, in terms of and using those methods to completely characterize a microorganism. We focused our efforts on Salmonella mainly, but also more lately, Coronabacter and Listeria monocytogenes. And these approaches are used in various applications, including genomics-based approaches for modern risk assessment of foodborne pathogens. What we're going to be talking about today, development of solutions to solve pathogen-specific problems, including development of diagnostics, antimicrobials, as well as bacterial subtyping. And more recently, combining metagenomics, artificial intelligence, and social media to develop next generation approaches to improve foodborne outbreak response. So Sam started off by giving a quick overview of why foodborne pathogens are such a major issue. As I said, in my lab, we focus mainly on Salmonella because it's globally, it causes a number of foodborne illnesses. You can see the numbers there, 80.3 million foodborne gastroenteritis cases each year and 150,000 deaths. And you can see the numbers in Canada and the US. So this is a pathogen that causes a significant amount of illness and deaths globally. Approximately a third of the deaths associated with foodborne pathogens each year around the world. And I just put up this slide to show that regardless of where we find Salmonella in the world, the serotypes that contaminate foods tend to be very similar. So there's 2,600 serotypes of Salmonella enterica, but only a few of those actually consistently cause illness around the world. And in this figure here from Cheng et al. 2019, we can see that the serotypes in the red boxes are the same around the world. So I have been leading a major project, which I call Salmonella Food Systomics. This is a major project funded by an agency called Genome Canada here in Canada. And it consists of a pan-Canadian international network, including researchers from universities across Canada, the US, federal government agencies here in Canada, including the Public Health Agency of Canada and Health Canada, in the US, including USDA, and as well in the Netherlands, as well as industrial partners. And so one of the things that we did was we developed a collection of isolates, currently 3,143 Salmonella isolates, consisting of approximately 250 serotypes, and we've sequenced all of them. So we have this biobank and the corresponding metadata and sequence data, which can be used to study a number of problems. And so in the bottom right of the screen, you can see what we've been focusing on at the farm level, development of a biochemical product. So one thing I should say is also in my lab, I really focus on the use of bacteriophages to solve problems. And so I'm highlighting that in this talk. So biocontrol product that we developed, and you can see the publication there. And then we'll be talking about diagnostics today, phage-based diagnostics. And then we've also developed subtyping methods using phages, prophages and their sequences within genomes for Salmonella. And you can see one of the publications there. The database is available at this link here at the bottom, where you can go and see the isolates and their sources. So this just shows us a phylogenetic tree of the core genomes of the isolates in the Salmonella collection, and I just highlight this to show the enormous diversity of Salmonella in terms of genomic sequence. And so you can see there's several clades. Salmonella enterica is the major subspecies. There's 6 subspecies of Salmonella enterica, and subspecies 1 at the right, at the left there, you can see forms 2 major clades. That's the major subspecies involved in pathogenesis, but you can also see there's other subspecies further out to the right. This figure at the top right is the same as the larger figure, but you'll notice in the larger figure, there's breaks in the arms, because if we didn't put the breaks in the arms would, these arms off to the right would go way off the screen. So this really shows enormous diversity. And this is important to understand when one is developing any type of intervention against Salmonella, whether it be a diagnostic or a control agent. It has to really be able to cover all of the diversity of isolates that we see. So this brings me to the topic, which we'll be talking about today, which is an assay, a reported bacteriophage or phage-based assay that we've validated. So just quickly, reported phage assay has been around for a while now, approximately 25 years or so. And they all have the same thing in common, which is that a reported gene is inserted into the phage chromosome genome, and then when it is expressed, or into the phage chromosome, excuse me, and when it is expressed, it produces some kind of signal. And that signal can be fluorescent in nature, it can be luminescent, it can be a colorimetric signal. We did some early work about 20 years ago with a colorimetric assay as well. And so that signal can then be read by the naked eye or some instrument like a luminometer or fluorometer. So when we talk about rapid pathogen detection, there's basically two types of assays that are in use commercially for detection of foodborne pathogens. First are molecular assays, including PCR, LAMP-based assays, and other assays like that. And then there's the affinity-based assays, so things like rapid antigen tests, ELISA-based assays, and so forth. And I've added here bacteriophage-based, because I think bacteriophage-based assays have some important advantages over the main other assays used to detect foodborne pathogens. And that includes the fact that they only detect live cells. So that's becoming more important. I think historically, in terms of foodborne pathogen detection, that was not seen as important, but it's becoming increasingly important. For example, the USDA FSIS is currently looking at developing methods for quantification of Salmonella in raw poultry as a way to decrease the presence of the pathogen in raw poultry products. And so bacteriophage-based assays represent potentially a rapid way to quantify bacteria, and I think will be useful there. And also useful for some of the other approaches that are targets that Dr. Liu mentioned, such as detection of antibiotic resistance and so forth. So the objectives of our work were to evaluate the diagnostic sensitivity and specificity of an assay called the phage-DX Salmonella assay, and to compare the analytical sensitivity of this assay to other assays, including the VIDAS SBT assay, which is an affinity-based assay, which uses phage tail components as part of its to capture the bacteria, followed by a monoclonal antibody to detect it. and a PCR assay called the BAX assay. And we did this in lettuce and ground turkey. So just a bit of a background about the phage DX assay. It's been recently developed by LabCorp and recently received AOAC approval. And the assay is based on the use of two phages that have been genetically modified to carry the nanoluke gene, which produces luciferase upon phage infection of viable salmonella cells, and that therefore produces relative light units, which can be detected in a luminometer. So to measure our diagnostic sensitivity, we used isolates from our Salpos collection that I recently showed, and we chose 107 isolates from that collection. I should mention that collection contains isolates from all over the world. So we really had a genomic diversity and geographical diversity, as well as diversity in terms of sources where those bacteria came, food sources, environmental sources, animal sources, and human clinical sources. So we chose a diverse number, 107 isolates from that collection, including Salmonella bongorii and Salmonella enterica isolates from both of those. And then in terms of diagnostic specificity, we evaluated against 199 isolates from non-Salmonella species, that were, most of which were closely associated to Salmonella and are within the Enterobacteriaceae. So to do this, we just took overnight pure cultures, and the 107 isolates consist of 58 serotypes of Salmonella, and we found that this assay could detect 97 of 107 of those isolates. And for specificity, same thing, overnight culture, approximately 10 to 9 CFUs per mil, and the phage DX assay correctly produced negative test results for 192 of the 199. So there were 7 false positives. So that means that our positive predictive value for the assay is 93.3%, and the negative predictive value, which is a measure of the assay to properly produce negative test results when it's a non-salmonella bacteria, is 95%. So we want to dive into these isolates that produce the false positive results. And it turns out that all of them are Citrobacter. So we had previously tested these Citrobacter isolates using the Vitis SBT assay. That's the bottom chart here. And you can see that there were also, this assay also misidentified Citrobacter as Salmonella. So the chart on the right shows how we isolated these Citrobacter. So these Citrobacter came from a large producer of fresh produce here in Canada, who had reported a large number of false positive assays. They were using the SBT assay, the Vitis SBT assay, and they reported a large number of false positives following a major rainstorm on their fresh produce. So the isolates were identified as Salmonella, presumptively, but the confirmatory test showed that they weren't Salmonella. So we took those enrichments, and we want to figure out what was in there that was causing the false positive results. So this is the approach we did that shows what we did on the right side. We took the enrichments, we reconfirmed that they did in fact test positive on the Vitis assay. If they did, we then serially diluted them tenfold and plated them in duplicate. This is step number 2. We then took the plate that had the highest number of isolated colonies, isolated all of those colonies in a single sample, and ran them on the Vitis. And then if that produced a positive result, we took the duplicate plate at the same dilution and individually tested each colony to determine which ones were giving the false positive. And so we were able to isolate a number of Citrobacter that way, and reconfirm that they did in fact in pure culture test positive on the SBT assay, and some of those, 7 of those 18, also tested positive on the phage DX assay. So this really, we really want to dive down more into into this and see what was going on with these Salmonella and Citrobacter. So what you're seeing here is a paper from our group about 2 years ago, from our consortium from 2 years ago, where we sequenced the Citrobacter and compared them to the sequences of the Salmonella that we already had. So on the left side of the screen, you see a maximum likelihood phylogenetic tree. And I should say that before we did this work, we actually did 16S RNA sequencing of these false positive Citrobacters. And when we create a tree from those, as well as members of the Enterobacteriaceae, including Salmonella, we saw that the Citrobacters did not separate. They did not form a distinct clade. So we want to drill down a bit deeper. So we sequenced the isolates, the whole genomes. And what you're seeing on the left-hand side is a maximum likelihood phylogenetic tree based on the genomic variations in the amino acid sequences of the core genomes of Salmonella and Citrobacter. And so what you can see is that two distinct clades were formed. This one at the top is Salmonella, and this one at the bottom is Citrobacter. So they're separated when we look at the core genome. However, when we did a bit deeper dive, we find that while they're separated, they're quite highly related. And that's what figure 2 shows. So this figure 2 on the right side of the screen is a network analysis based on shared genes. And so connectivity between strains indicates the presence of common gene families. So if you, for example, in figure 3, you see, or C, you see lines between these two groups of isolates. And so that really represents shared gene families. But what I wanted to highlight is this A here. And I should say before we get to A that the different figures denote different percentages of thresholds of the shared genes. So figure A is 26%, B is 27%, C is 29%, and D is 34%. And you can see as we increase the thresholds, there's less of a connection between these isolates. But I really want to focus on figure A, which shows all of the isolates. The blue ones are Salmonella, and the other colors are Citrobacter. And so you can see that in figure A, 26% connectivity, all of the strains, regardless of whether they're Salmonella or Citrobacter, form one community of interconnected members. And so this really corresponds to what we also found with the core genomes, which showed about 30% of core genomes in the Salmonella and these Citrobacter isolates are shared. And so keeping in mind that phage-based assay, the first step is binding to the bacteria, recognizing receptors on the cell surface. The SBT assay is an affinity-based assay. So again, the first step is binding. It's likely that these Citrobacter isolates share common protein homology on the cell surface with their Salmonella counterparts. And this has previously been shown for various isolates, but what we have shown here is that it's strain dependent. It's not, so for example, Citrobacter freundii, not all Citrobacter freundii will demonstrate this. It is strain dependent. So getting into the work on analytical sensitivity, we first tested lettuce, and this slide shows the procedure. So we use an AOAC procedure for fractional inoculation. These are 25-gram samples that we inoculated with a 3-strain cocktail of Salmonella, including isolates from serotypes St. Paul, Newport, and monophasic Salmonella. We, after inoculation, cold stressed the samples for 48 hours, and then we did 30 replicates. We tested 30 replicates, where 20 of those were fractionally inoculated, 5 were inoculated with a high concentration of Salmonella, and 5 were uninoculated. And so the low level or fractional inoculation was estimated by MPN to be 0.59 CFUs per 25 grams. And the high level inoculation, about a log higher, was estimated at 2.97. And so what we did was, following the cold stress period, we incubated the samples in the 3 assays. The phase GX assay is approved, AOAC approved for 7-hour and 18-hour detection, and we did the Vitis SBT at 18 hours, that's per its AOC approval, and the BATS PCR system at 24 hours. And then we looked at the results. We also did serological testing of colonies from the enriched samples to, to confirm that there were salmonella. And we also compared these tests to a reference method, the Health Canada reference method, the official Canadian method used to confirm the presence of salmonella in foods, and that's very similar to the FDA BAM method in the U.S. And I just want to highlight also that because at least in this sample we used different enrichments because we followed the manufacturer's instructions. This was an unpaired study, so the phage DX assay and the SBT assay we used different enrichments, so it was an unpaired study, but we did pair the BATS PCR and the reference method in this study because we use a similar enrichment, the same enrichment. So here we see the results. So these graphs show the results. So what you're seeing on the x-axis is a calculated MPN for 25 grams, and on the y-axis is the probability of detection. And so this point here, this represents the 5 uninoculated samples that did not have salmonella. So and I should say that the blue is the reference method and the red line is the candidate method, in this case the phage DX assay. And so the 5 uninoculated samples are here. None of them had salmonella, so neither the reference or the candidate method detected that. This next set of samples, these would be the 20 fractionally inoculated samples. And we can see that based on the reference method, 40% of those 20 samples had salmonella. The phage DX assay produced results that showed 50% were positive of the fractional isolates, but based on the error bars, these two results are the same. And then finally, this value at the top here was the high inoculated samples, which were all positive by both methods. And so this graph on the left is at 7 hours and at 18 hours. So we can see that the phage DX assay agreed very well with the reference method. And then we also see that when we did the same thing with the Vitis SBT and the BATS PCR, that the results were similar to the phage DX assay. So in other words, all three rapid methods agreed very well with the reference method in lettuce. So then for ground turkey, again, we did AOAC procedure. This time we used still a 3 salmonella cocktail, but different serotypes: Hadar, Schwarzenbrand, and Heidelberg. These were, as with the lettuce, we chose serotypes that had been previously implicated in outbreaks. And this time we tested 375 grams, which we cold stressed for 48 hours. Again, the same 30 replicates. I should also mention that in terms of we enriched instead of doing an enrichment of 1 in 10 for both the lettuce and turkey, we did 1 in 4. This is because there's an interest in the food testing industry in terms of reducing the amount of volume of these enrichments. And certainly if you're testing 375 gram samples, to do a 1 in 10 becomes quickly not feasible because of the large volumes and the need for large incubators and so forth. So our dilutions were 1 in 4. We did this in this work. We did this study a little bit differently than the last one, in that we tested the phage DX assay at 7 and 18 hours as per its AOC approval, but we also did additional time points because we want to see how the assay would behave and compare to the other assays between the time points of 7 and 18 hours. And so, as with the last, we did MPN to estimate the low and high level contamination. So the low level here was estimated to be 1.86 CFUs per 375 grams, and the high level was 4.74 CFUs per 375 grams. So here we see the results. What we're seeing here is the results. This is two charts, one at 7 hours and 9 hours post-enrichment. And you can see there's several columns: the phage GX assay, the Vitis SBT assay, the BAX PCR assay, the culture confirmation official Health Canada method, and you can see the CFUs that were actually present at the time of the assay. So this gives us the detection limits that the assays are able to detect. So what I want to show is that at 7 hours, the phage GX assay was able to detect all of the samples that were subsequently confirmed by the culture confirmation method to be positive. In contrast, the Vitis only detected a few, and the BAX PCR only detected one of the samples. At 9 hours, we see the phage GX assay again detecting everything that the culture confirmation did. The Vitis assay, the same. The BAX PCR assay, a few more samples, but still not all of them. And it took until 11 hours for the BAX PCR assay to detect all the samples. So what this shows is that the phage GX assay is very sensitive. If we go back to this slide, we can see that it can detect as few as 10 to the 2 cells, which we also subsequently showed. I didn't present this, but in pure culture, it was able to do that for a number of salmonella isolates as well. So it's very sensitive, and it produces results faster than the two assays, at least in Turkey, at 375 grams in this study. So to summarize, Salmonella enterica is a foodborne pathogen of global importance. It's been implicated in many outbreaks linked to lead in Turkey, among many other foods. We've evaluated the phage GX assay for diagnostic sensitivity and specificity, as well as analytical sensitivity. And when compared to existing commercial assays, the assay performed well and provided accurate results within a faster time period in lettuce or ground turkey than the other assays. Our future work is aimed at assessing this assay in difficult to test food matrices, including spices. Dr. Liu talked about the presence of the antimicrobial compounds in garlic, for example. Those compounds are also present in other produce from which spices are derived, like onions. And that can actually inhibit, and has been known to well inhibit commercial testing assays. And actually, because it can, not only can the compounds inhibit PCR assays, for example, but they can also inactivate the bacteria and stop and kill them during enrichment. So we want to evaluate this assay in those matrices, including and also dry foods like pet food, which has been implicated in a number of salmonella outbreaks. So I'd just like to acknowledge the funding for this project and the people who worked on it, including at the Agricultural and Food Laboratory at the University of Guelph, which is an accredited testing laboratory, and the people in my lab that worked on this. And with that, I think we have some time for questions. Thank you very much.